Hello, I'm François Chaumont from UC Louvain in Belgium. First of all, I would like to thank Kathleen Bird for giving me the opportunity to speak in the frame of this workshop. It's always great to talk about our favorite topic. The general title of my talk, Aquaporin Function and Regulation, from Molecular and Cellular Characterization to Plant Physiology, actually reflects the work we are doing in my lab since many years. So we were asked to follow a general structure in relation to the talk. What is your current research focus? What aquaporin research discovery do you find most exciting? What unsolved mystery related to aquaporins would you most like to see solved? Actually, all these questions are linked to each other, and I will address them simultaneously, notably by taking example in the research we are currently doing, believing that, modestly, we contribute to unsolve some mysteries related to aquaporins. Beyond the discovery of the first plant water channel activity in 1993 that changes our view of plant-water relation, the first puzzling observation is that aquaporins constitute a large and divergent protein family in plants. This is illustrated here with the maize aquaporins that cluster in 4 subfamilies, as in other monocots, and the Brassicaceae, including Arabidopsis thaliana. A 5th subfamily is found in other vascular plants. In total, we have a large number of aquaporin isoforms. Of course, it is not unusual to have a proliferation in the copy number of critical genes in plants by frequent whole genome or tandem duplication events. But this definitely complexifies the story regarding aquaporins. Indeed, we have a huge diversity in expression pattern, membrane localization, substrate specificity, and of course, physiological roles, but also in the regulatory mechanisms. Let me illustrate this in this slide, focusing on plasma membrane intrinsic protein, or PIPs. PIPs are regulated at different levels that start by the transcription and then the translation in the endoplasmic reticulum membrane, where we can see that some PIPs can form heterodetramers. Then the protein moves through the circular pathway to reach the plasma membrane. Once in the plasma membrane, the channels can be opened or closed through opening and closure by post-translational modifications such as phosphorylation or protonation. All these regulatory processes depend on environmental cues or developmental stages and involve phytohormones or signaling molecules and the circadian clock. In addition, PIP aquaporins facilitate the diffusion of water, but also some of them can diffuse urea, hydrogen peroxide, carbon dioxide, and even cations. These findings raise the following fundamental question. Why are there so many aquaporin isoforms and so many levels of regulation? And how all these regulatory processes occur and interact with each other in planta? We can even ask this question, would plants be able to behave the same way with less aquaporin isoform and regulatory mechanisms? Let me show you how we tackle part of this question. So I will start by a project we are working on to decipher aquaporin expression variability in maize genotypes and identify actors involved in this regulation. To address this question, we perform an EQTL study and characterize cis and trans-actin factors regulating PIP aquaporins. This work started several years ago in the frame of an European crops project. We get access to a large panel of more than 250 maize genotypes that were extensively genotyped with around 1 million of SNP markers. And they were also precisely phenotyped in various conditions, leading to a large amount of data. We also benefited from the experts that studied the population structure and set up the models. We perform a GWAS study and map the EQTL that allow us to identify some transcriptional regulators. Actually, We measure by qPCR the expression of 5 PIP genes in well-watered conditions in 2 zones of the leaf, the elongation zone and the mature zone, in which specific physiological processes occur, such as growth and photosynthesis, that could be related to aquaporin activity. So here are the EQTL for the 5 pips in the elongation and mature zones. These are actually Manhattan plots, a view from the top, the highest peak being closer and therefore bigger. We can observe that in most cases, the largest EQTLs were located very close to the PIP genes of interest, while other EQTLs are spread throughout the chromosomes. In local EQTLs, we expect to have cis EQTL that influence the expression of the PIP gene and can consist, for instance, of sequence differences in the promoter regions, in sites for small interferon RNA binding, or in an enhancer. The distant EQTLs are trans-EQTL that can be genes that encode transcription factors, epigenetic-related enzymes, or sequences for small interferon RNA. Let's consider now the local EQTL for PIP25 in the mature zone. The allele at this position defines 2 distinct haplotypes in the population, colored in green and red. Among the genotypes, we use B73 and PH207 as representatives of the green and red haplotypes. The B73 allele was associated to a higher PIP25 expression compared to the red haplotypes. Comparing the sequences of the 2 haplotypes, we observe that they are well conserved except for several insertions. So we observed that in PH207 we have several small insertions close to the PIP25 genes, and we were quite interested by this one that included a mild transposon. Mild meaning miniature inverted repeat transposable element. We selected a few genotypes from both groups in order to look at the conservation of the mild containing insertion, and we saw that the transposon presence was correlated quite well with the green haplotype. With the red haplotype. So it's present in the red haplotypes where the expression of PIP25 is lower and is absent in the green haplotype. To evaluate the effect of the mild included insertion on PIP25 promoter activity, we designed a reporter gene assay in which the different PIP25 promoter were fused to the M genus. So we have here in green the PIP25 promoter from the B73 and here the promoter from PH207. In addition, we added the insertion containing the transposon in the sequence of P73, or we remove this sequence from the PH207 sequence. We measure the fluorescence of M Venus, and we normalize the data with the M cherry fluorescence coming from 35S M cherry transgene. And we can definitely observe on this panel that the level of expression is significantly lower when mild transposon is present compared to the expression when the transposon is absent, like here in the B73 PIP25 promoter or in the PH207 promoter without the insertion. So it means that transposon, but also maybe the other insertion, can contribute to the variation of PIP gene expression. Using EQTL's data, we have also identified a transcription factor we are currently characterizing that seems to also affect PIP25 promoter activity. Altogether, GWAS and EQTLs are powerful tools to decipher the variability of aquaporin gene expression and identify characterized molecular regulators. I would like now to talk about another project we are working on that should allow us to better understand both the trafficking and the activity specificity of the PIP aquaporin. The model used is the stomatal complexes, and actually we focus on the role of PIP1 aquaporins. So we previously showed that different PIP aquaporins are expressed in maize stomatal complexes, but what is that the fact that more than 85% of the PIP expression is due to PIP1 isoforms. So the PIPs can be divided in two groups, the PIP1 and the PIP2, and both proteins behave differently. And actually, PIP1s and PIP2 physically interact within heterotetramers to regulate the trafficking of PIP1s to the plasma membrane and the channel activity. So to study the role of the PIPs in the stomatal complexes, we decided to modify the expression of these proteins. So, and here I'm going to show you data that concern PIP1/6 that is specifically expressed in the stomatal complexes during the day. So we produce transgenic maize lines overexpressing PIP1/6, and we look at the stomatal behavior. We can see here that the stomatal conductance of maize lines overexpressing in PIP2/5 is higher compared to the wild-type plants. Interestingly, we also observed that the closure of the stomata after switching off the light is faster for the PIP1/6 overexpressing plant compared to the wild type. And similar data have been obtained after additional high carbon dioxide concentration or after treating the stomatal complexes with PEA. So why do we observe this kind of phenotype? Why do stomata close faster in PIP1/6 overexpressing lines than in wild-type plants? So we previously showed that PIP1/6 can act as a water channel when co-expressed with PIP2/1 in xenopus oocyte. We can see that both proteins interact to increase the osmotic water permeability of the oocyte membrane. So it means that tubular pressure in gut cells may change faster in PIP1/6 overexpressing plants than in wild-type plants. But we also show that PIP1/6 might facilitate carbon dioxide diffusion after its expression in the yeast Saccharomyces cerevisiae. So carbon dioxide concentration inside the gut cells may be higher in PIP1/6 overexpressing plant than in wild-type plant. And finally, we also observe that ABA-induced hydrogen peroxide accumulation is faster in PIP2/6 overexpressing plants, and in gut cells in particular, as observed here, compared to the wild type. So ABA and H2O2 signaling may be more efficient in PIP1/6 overexpressing plants. Does one PIP aquaporin doing everything in the gut cells? Actually, this was also shown in our Arabidopistariana with PIP2/1. PIP2/1 was shown to fulfill different roles by facilitating the diffusion of H2O2, water, and carbon dioxide, and all these processes leading to the closure of stomata. But then why are there so many aquaporin isoforms expressed in stomatal complexes? And why are PIP1s much more abundant? This is quite puzzling because we know that PIP1s need PIP2 to reach the plasma membrane. How PIP1s reach the plasma membrane if we have so few PIP2 proteins? And finally, how do all these channel activities and regulatory processes are integrated? These are the questions we are going to address in the future in my laboratory. So I will stop here by acknowledging the people who perform the work. So the data about the stomatal complexes were obtained by Robert Heinen, Thomas Milliet, and Lei Ding. The EQTL study was performed by Laurie Mestrio, who will defend her thesis this November. Also thank the different collaborators in Belgium, in Ghent for the maze transformational platform, but also in France for the EQTL analysis in Montpellier and Le Boulon. Thank you very much for your attention.